And the number of methionines is 3. Okay, so they all have to be internal, and they can't be on the C terminus. Does that make sense? So let me put that like that. So 18, right? And so I've got, this is my N terminus, this is my C terminus, and that's what we said. How to read it is from the N to the C, which means the methionine can't be here. The methionine is here. So let me put methionine here. Methionine here. So when I cut with cyanide and bromide, I cut here and here. So that would give me 3 peptides. Right? So I say I'm going to get 4. So it can't be at the C terminus. If I put methionine here, I only get 3 peptides. So they all have to be internal, and I do need to put 1 here. Okay? Does that make sense? Because then only will I get 1, 2, 3, 4. So the answer is false there. Any question on this one? Okay, so the next one is I give you 7. Right, and probably the easiest is this one. That'll be the easiest to start with, and that tells you that alanine is right after the first ring. Okay, so that's where that has to be. Then trypsin, where would trypsin be? Because it cleaves at arginine and lysine. So, and I'm telling you, the R and the A and the V are together, not the order, right? Because I have a comma, that means I know they're together, but not the order. So you gotta figure out the order. And then the rest is R, S, Y. So it's the fact that I have A here means I either have to put the R here or V here. Okay, so I've got to put my R here. Right, there's no other place it can be, because if I cut with trypsin, I'm going to get my AVR. If I put the R in the second position, I would have said the combination would have been an A and a V together. Right? Okay, so that one's there for sure. Then, look at the R here. It's combined together with the S and the Y. So the only logical place it can be, the only logical place that it can be is here. With this clue, right? I can't put it here, I can't put it there. Because it has to be together, R, S, and Y have to be together. If I put the R here, then it would begin to cut right after this, 4th. And then of course last is easy because I already told you R, A, B, and Y have to be together. So Y has to be, tyrosine has to be the 4th position. You guys have any questions about this? Okay, so the back of your page has some extra problems that I want you to work on. I'm not going to be working on those with you, but feel free to come to office hours if you're still having difficulty with these types of problems. Let's move on. I did give out the hemoglobin worksheet. Probably, maybe, let's see how far we get through it. If we don't, we'll come back on Tuesday and work on it. So now we're moving on to the next biomolecule, which is carbohydrates. And just because we're moving on doesn't mean we won't see protein. We'll see protein, we'll see amino acids along the way. But this is a 1-chapter section on carbohydrates. And it's an important chapter because I am going to be using some of the material from here that needs to be used for Biochem 2. So the lipid chapter are a lot of memorization. And so it's important for you guys to come here because I tell you exactly what you need to study. Alright, and I mark it down, I put underlines on things that you need to know, so pay attention to those and go back to your slides to look at them, because there's a lot of information here. We're going to look at monosaccharides, we're going to look at monosaccharides, which are the simplest sugars, and of course then we put the two monosaccharides together and get disaccharides, and there are 3 of those that you need to know. Polysaccharides, very important, is glycogen. I need you to know the structure of glycogen, because I'm going to use that for biochem too. Okay, to synthesize glycogen and to break down glycogen. Glycogen is a storage form of glucose, and starch is also the same. Starch is going to be divided up into 2, amylose and amylopectin, and those are found in plants. And that too is a storage for glucose. And then we've got cellulose, which has a different function, which is for structural purpose. So in all of these, of course, you need to know what they look like, chemistry, right? The structure-wise. You need to know how they're branched or unbranched, and what their functions are. So really memorize it. There's really nothing fancy about it, just memorize it. And retain it. That's the key. Retaining it. So this semester, last semester, Biochem 1 and 2, I gave them a question on glycogen. Most of them got it right in Biochem 1. Then I asked the same question in Biochem 2. It hadn't been retained as well as I had thought. So retain the information here, particularly those ones that I'm pointing at. Because again, I repeat it and review it in Biochem 1 at the first lecture, but it really needs to be retained. Then we talk about those proteins that have carbohydrate groups attached to them. Those include glycoproteins, proteoglycans, and mucins, and you just compare and contrast between them. We're going to look at some of the clinical insights. There's lots of different clinical insights there, and that's really the most interesting part. Not just memorizing it, but actually seeing why we study glycobiology. Okay, so we're going to look at that. Glycosylation is a term that's used as post-translational modifications. So protein is made, and then carbohydrates get attached to it, and that process is called glycosylation. All right. We're going to look at penicillin. It's not part of the chapter 10 in the carbohydrate chapter. I talk about it because, one, it has carbohydrates, all right, the peptidoglycan, and it has amino acids, and it has connections to penicillin. So we talk about looking at structure this semester, and then we dive into a little bit more when we look at it as a competitor next semester. And then Tamiflu. How does Tamiflu work? Okay, let's look at what the functions are. Carbohydrates, energy. So that's really what metabolism is going to be about. Taking the sugar molecule, whether it's glucose, fructose, galactose, glycogen, we're going to break it down. And from breaking it down, we're going to oxidize these individual saccharides and then get energy out of it in the form of ATP. Next is supplying the carbon for the synthesis of cell components, and that's where we talk about the cell wall and the presence of the peptidoglycan, and that's where I get connected up to penicillin. Serve as a form of stored chemical energy. For example, the liver, for instance, which is a sensor for blood glucose level. If there's an excess of blood glucose, then the liver will take it up, and that glucose is stored in the form of glycogen, which is also a stored form of energy. Form parts of the structures of the cells and tissues. Okay, so actually when you look at a little bit more about the membrane transport chapter, you'll notice that the lipid bilayer has carbohydrates attached to proteins, and they are important in terms of communications. And we'll talk a little bit about that at the end. I'm just giving you some examples. This is not the only ones you need to know. Just examples. Monosaccharides are glucose and fructose. Okay, those are important, and galactose Those are important because we do talk about them in Biochem 2, how to metabolize glucose and how to metabolize fructose and galactose, which we just did. I just did with the class this morning. Disaccharides, 2 monosaccharides together. Example would be sucrose. Oligosaccharides, we're not going to talk about oligosaccharides, so at least 10 sugar groups linked together. So I've given you an example of those. And then we are going to talk about polysaccharides, examples, glycogen, starch, and cellulose. First, let's look at the chemistry behind what the structure of a sugar molecule looks like. You can classify them based on whether they're an aldehyde group or a ketone group. So here's your aldehyde group, and here's your ketone group, right? So they, obviously you're seeing the same atoms, carbon, hydrogen, oxygen, also in carbohydrates, just like you did with amino acids. So these can be classified based on whether they belong to the aldehyde family or the ketone family, and they can be categorized by the number of carbons. So this one is a simple sugar. Dihydroxyacetone and glyceraldehyde are simple sugars, and dihydroxyacetone is a ketose and can isomerize to glyceraldehyde, and that's an aldose family. Right, and it's 3 carbons, and it's the smallest and it's the simplest, and from there you can derive the different sugars. So this is, these two you'll see, dihydroxyacetone and glyceraldehyde, in terms of a phosphorylated form when we actually break down glucose next semester. And you will see them going from a 6-carbon glucose molecule to 2,3 carbon. So those 2,3 carbon molecules are dihydroxyacetone and glyceraldehyde versions. So they are the simplest sugars. So like I said, they isomerize. Glyceraldehyde isomerizes to dihydroxyacetone, so you can go from an aldose to a ketose and vice versa. And that's important because when you guys go into the lab, you'll have a sugar, which is a ketose, which I'll tell you. And when you take that ketose, it actually isomerizes back to an aldose, and you can actually see a color change and state that it's a reducing sugar. So these terms should be reviewed terms, so I'll go through them. So isomers have the same molecular formula, but obviously different structures. You can have constitutional isomers and stereoisomers. Constitutional isomers, they are different arrangements, like we just talked about with the example of glyceraldehyde and dihydroxyacetone. They have the same molecular weight, but just the arrangement is different of the atoms. Stereoisomers can be divided up because the order in which they are arranged is different, and they can be divided up into enantiomers and diastereomers. Enantiomers are non-superimposable, mirror images of each other, and you can designate them as a D and an L. And then the diastereomers are not mirror images. Those can be divided up further through epimers and anomers, and epimers differ as one of those several asymmetric carbons, and I just outlined in star what that asymmetric carbon is. And then the anomers, of course, you can also have it in the alpha form or the beta form, right? The alpha form is the OH is on the bottom, and the beta form is OH is on the top. So that again is different as a new asymmetric carbon. So let's classify them. You have aldose that have aldehyde to them, ketose that are sugars that have ketose groups to them. Then you can classify them based on number of carbons. So triose has 3 carbon, tetrose has 4 carbons, pentose has 5, and hexose has 6 carbons. This is no longer in the version 4 edition, but I really like to use this because number one, I can actually see where they're coming from. Glyceraldehyde is a 3-carbon sugar, and they belong to the aldehyde family. And you know it's an aldehyde right there. Right? So this all belongs in the aldehyde family. So the ones underlined are the ones you need to know. You need to know the number of carbons and which family they belong to. So glyceraldehyde, ribose, is important because we're going to be talking about it in terms of the nucleic acid. So that one's important. So that's a pentose, and it belongs to the aldehyde family. Okay, so there's your 5 carbon, so it's a pentose, and it belongs to the aldehyde family. You also need to know glucose, obviously, because we're going to be talking about it next semester. Galactose, same thing, and mannose. That is also belongs to the aldehyde family, and there are 6 carbons there. Alright? So pretty straightforward. So this slide is all aldoses, and I've told you which ones you need to know. Same thing, the ones underlined are important, and this is a ketose family. Dihydroxyacetone is important because obviously it's going to be a metabolite in 322, so I would like you to know that it belongs to the ketose family and has 3 carbons, and it can isomerize with the glyceraldehyde that I just talked about. So this one isomerizes to this one, right? Glyceraldehyde that I just showed you a picture of right here. Okay, so they do isomerize. One belongs to the aldehyde family, the other one belongs to the ketose family. And then fructose is important. Fructose is a 6 carbon, so it's a hexose, and it belongs to the ketone family, so we call it ketone hexose. Alright, and that one's important because obviously we're going to metabolize that one too, both in the adipose tissue and the liver next semester. Alright, so that's pretty straightforward. So just come and memorize it and get that question right. So pentoses and hexoses of the aldoses and ketoses can exist both as a hemiacetal and hemiketal. So this too should be a review, but sugars can also do this because they do have the alcohol group, and they can attack the aldehyde or the ketose part of the molecule. So when you want to make hemiacetal, the alcohol group attacks the aldehyde, and you can get a hemiacetal. The same thing. The alcohol group can attack a ketone, and you get a hemiketal. You can take the same principle and use it and apply it to sugars. Alright, so I'm giving you an example here of a sugar, glucose, that at the carbon 5, where's my carbon? Yeah, carbon 5, attacks the aldehyde group of the sugar. Remember, glucose belongs to the aldehyde family, and it's 6 carbons. So when it attacks, it can form into a ring structure, and these two, either the alpha or the beta, looks like a parent, right? So they call it a parenose. That's where the name comes from. And then you can do the same thing with fructose, which belongs to the ketose family. And that being a 6 carbon too, just like glucose, but the only difference is it's a ketose family. And again, you have an alcohol group attacking the ketone group, and you form a cyclic form of the fructose, and because this structure looks like a furan, it's called a furanose. We really don't know, but it looks like nature favors the pyranose family and the furan family. I'm just giving you an example of when they do these hemiacetal and hemiketal reactions. Any of these sugars, glucose, mannose, ribose, or fructose, whether it's 5 carbon or 6 carbon, it's only the 5 and the 6 carbon sugars that does this. And so when it does it, you form a most unique pyranose family. And it likes to exist in the chair form. And this structure-wise likes to exist in the chair because look at the steric unions between these two groups. Okay, so that because of that, it likes to exist in the chair. This is a lab part, which is nice to have because this is something that you're going to do with the carbohydrates. You take sugars, different sugars, and you see if they're reducing sugars. And so those sugars that react with these oxidizing agent, copper, if they get reduced, then that particular sugar is called a reducing sugar. Only the sugar groups that have aldehydes will be reduced, and so they are called the reducing sugars. Okay? And those sugars that cannot be reduced, obviously, are called non-reducing sugars. And you just see a color change. Alright, so it's more of a qualitative experiment that you do. It starts out blue, and then once you add reducing agents, it turns into a dark brick color, and that's your reducing sugar right there. This reagent that you're going to use has a base in it, and so when you add a base to it, you can actually isomerize. So you'll be given fructose. And I just told you that aldehyde family get reduced, they're exposed, so they'll get reduced, so they're reducing sugars. But you'll also be given fructose. And fructose will change color. And you'll be asked, why does it change color even though it is a ketose? Because fructose with the base can isomerize to glucose, and then it gets reduced. Because now it has become an aldehyde family. Okay, so there are sugars that get modified, just like proteins get modified, sugars can also get modified. Some of the ones that you will see next semester are glucose 6-phosphate. So when we begin glycolysis and take glucose and phosphorylate it, and all sugars need to get phosphorylated, that's usually the first step. Because once they're inside of the cell through the transporters, they need to get stuck in the cell for glycolysis to occur, so they can't go backwards. So in order to prevent that glucose from going backwards out of the cell, the first step is going to be phosphorylation. And so glucose is modified through phosphorylation. So it becomes glucose 6-phosphate. I have told you the stage 1 of the glycolytic pathway ends when we take the 6-carbon sugar and split it up into 2. Those 2, one of them will be dihydroxyacetone phosphate, we just talked about, and then the glyceraldehyde 3-phosphate. Alright, so you'll get 2 3-carbon sugars. So again, another sugar that's phosphorylated and modified. Today, we're going to see sialic acid. This sugar is important because the viruses have these sugars in their cell surface membrane, and this is how Tamiflu will work, preventing the deattachment of the virus from the cell membrane. Other ways to get modified. Here's a single monosaccharide at the anomeric carbon. It gets modified. And when it's attached to the oxygen here, it's called O-glycosidic bond. Okay? So O because there's an oxygen there. Glycosidic because it's a bond with the glucose or any sugar. Doesn't necessarily have to be glucose, it can be any sugar. Okay, so that's called an O-glycosidic bond. Another name is N-glycosidic bond. So with N-glycosidic bond, there's your ribose. Attached to the ribose is a base. And because the base is attached to a sugar, it's still glycosidic. There's a sugar there, so it's the glyco. And it's attached to the N, nitrogen. So it's an N-glycosidic bond. Okay, so you can have O-glycosidic bonds or N-glycosidic bonds. Any questions on monosaccharides, what you need to know? Okay. Disaccharides, linkage of 2 sugars together. And obviously all of those saccharides can be up to 10 sugars attached together, 2 or more, through glycosidic bonds. Let's look at bonds and naming and what you need to know for those. So here are 2 sugars. It can be anything, and you'll see 3 that I need you to know. So when they link up through an oxygen here, it's called an O-glycosidic bond. Okay, so there's sugar 1, sugar 2, and they're linked alpha 1, and alpha 4, carbon 1 and carbon 4. So it's an alpha 1,4 glycosidic bond. Alright, and it's O-linked. Just like I told you, there are N-linked, there's also O-linked. Okay, so oligosaccharides. I'm going to talk about oligosaccharides with not really anything sugar-wise you need to know, but I'm going to talk about it in terms of how you connect it up. And oligosaccharide, like I said, is 2 or more. But the enzyme that you use to link 2 or more is called glycosyltransferase. Okay, so really remember this one, because when we go to 322, we talk about how the deficiency of one of these glycosyltransferase can result in galactosemia and then eventually can lead to cataracts. So the monosaccharide substrate for glycosyltransferase is an activated form. A lot of the sugars that you have to link together to either create a disaccharide or oligosaccharide has to be activated, because you want the reaction to move forward, so input of energy needs to go in. So they will be in the form of UDP. So we'll talk a little bit more extra next semester about these linkages, but I want you to be aware that you do need to put some form of energy into the molecule so the reaction can go forward. So this is just an example, and you can use this for creating not only disaccharides, but oligosaccharides. It's just a linkages. And there's plenty of glycosyltransferase enzymes out there, very specific for the type of sugar that is going to link. So anyways, the glycosyltransferase takes a sugar, and an activated form of a sugar, in this case, they're showing you UDP glucose. And it's just a transferase, so it'll transfer. So the UDP molecule will attach itself, and the sugar group, or the hydrogen, the hydrogen group will get attached to the molecule UDP, creating back to UDP, releasing it, and then you have a sugar molecule attached to another sugar molecule here. So X is the sugar. Right? So that's your disaccharide. So again, you have an O link, alpha 1,4 linkages. Okay? So the X here is a sugar, all right, and it's just transferring over one of the sugars to this other sugar. And you create that disaccharide. And it continues. Again, you can add another UDP, fructose, glucose, whatever it is, and then you can keep linking it to create this oligosaccharides. Okay, so these are the sugars you need to know, disaccharides. Alright, so sucrose. What do I need you to know? I need you to know that sucrose is a combination of glucose and fructose. Lactose, if there's milk sugar, I need you to know that it is a combination of galactose and glucose. And maltose is two glucose molecules together. Alright, so these are the ones, really particularly lactose we'll be looking at, and then galactose we'll be looking at next semester. So I do need you to know the combinations of these. Okay, I don't need you to be aware whether it's a pyranose family or a furanose family or alpha or beta or gamma. I just need you to know the combinations.
Will you repeat those combinations?
Sure. Sucrose is glucose plus fructose. Lactose is a combination of galactose and glucose. Maltose is a combination of glucose with another glucose. Alright, any questions on disaccharides? Okay, let's look at polysaccharides. We've got two. We've got ones that are stored and ones that are important for structural purposes. So for storage, glycogen, right, is a storage molecule of sugar that is specifically glucose. So it's all strings of glucose attached together with glycogen. Starch in the plants can be divided up into amylose and amylopectin. And again, that's also all glucose molecules together. Because they are linking together, they are polymers, and because they're the same sugar, we call them a homopolymer. Structural group, cellulose. The ones that are highlighted in red are the ones that I'm going to talk about today, but there are others. Cellulose structural form is in plants, and then chitin is the structural form of polysaccharide found in insects, and then polysaccharides of the bacterial cell wall is something that I put in there because we're going to talk about penicillin and how that affects it. The bacterial cell wall synthesis. Glycogen versus starch, both of them can be digested by the enzyme amylase. So remember, glycogen and starch are homopolymers of glucose. Okay, all glucose molecules attached together. and so they can be broken down by the enzyme amylase. Okay, so glucose is the storage form of glucose, and where are they stored? They are stored in the liver. So liver has a lot of different functions in metabolism. One of the things is obviously acting as a glucose sensor and taking up the glucose when the glucose levels are high in the blood, or releasing it when it's low. But they are stored in the form of glycogen. In plants, there are my plants, the potatoes and the chloroplast, that's where your starch is going to be located, and that again is a storage of glucose. More memorization, nothing fancy there. Glycogen is a storage form of glucose. They are connected. Very important how they're connected, because we're going to synthesize this next year and then degrade next year as well. They are linked by alpha 1,4 bonds, glycosidic bonds. And every 10 or 12 units away of the glucose, you'll see a branching, an alpha 1,6. Okay, so this is what it looks like structurally. You have alpha 1,4, glycosidic bond. Here's glucose 1, glucose 2, linked together through alpha 1,4. And then about 10 or 12 units away, you'll see a branching. And you'll have alpha 1,6. Because they're different, because it's alpha 1,4 and alpha 1,6, when we start synthesizing it or even degrading it, you have different enzymes degrading the alpha 1,4 and the alpha 1,6. Okay? Totally different. Now we move on to starch. Starch has two, amylose and amylopectin. The one with the shorter name is very simple. It's just alpha 1,4 linkages, all alpha 1,4. Amylopectin, again another storage, but one the longer name has both alpha 1,4 and alpha 1,6. Okay, so, but this one is a little different from glycogen, because instead of the 10 to 12 units away, you'll start branching every 30 units away. That's the only difference. So amylopectin is going to be very similar to how a glycogen looks, except for where it branches and when it branches. Okay, so just to give you a visual aid of how this looks like. So here's your amylopectin. It's a long name, branching, more complex, right? So here you have the linkages. I can even use this one. So here's the linkages of alpha 1,4, and every 30 units away it starts to branch. So you see a lot of branching here going on here. And it doesn't necessarily have to be one sugar branching. You can have multiple sugars branching. Okay, so you can have branching here at alpha 1,6 and then continuation of an alpha 1,4 linkages. Cellulose is a little bit different because it is for structural purposes. So it's not a storage form for plants. So we got amylase and amylopectin, which are for storage of glucose. Within the plant, cellulose is for structural purpose. It all has, it's also a homopolymer, so that means it all has glucose attached to it. But instead of alpha 1,4, it has a beta 1,4. Alright? This is a little bit of hand waving of why they think cellulose is more structural versus that of starch or glycogen. If they look at the structure of this starch and glycogen are more hollow in nature, so they're able to compact and include more glucose. And this is more stacked up. Cellulose is more stacked up, so it's for structural purposes. Okay, so that's what you need to know about the polysaccharides. Right, just make a table for yourself and memorize it. All right, so glycobiology keeps moving on. So glycobiology is not just about just knowing about the monosaccharides and the disaccharides and the polysaccharides. It's actually using proteins that have gone through post-translational modification by having sugar groups attached to it. So proteins that have sugar group attached to it are called glycoproteins. So those can be divided up into simply glycoprotein because that was what was first discovered, so they named it. And then they began to classify it based on really what types of carbohydrates were attached to the proteins. They have a specific one called proteoglycan, and there's others like mucins. But obviously this is not the only three, there's obviously more to it. Okay, so where do they bind? Not all of these places, not all of the serine, not all of the asparagine is where the glucose binds or the carbohydrate binds. Okay? Not every serine, not every asparagine is going to have a carbohydrate group attached to it. But there could be. And it's been seen that in some proteins that have serine, there's a carbohydrate group attached to it. Okay? So here we have in blue a peptide, right? So here's your alpha carbon, carboxylic acid, amino group, hydrogen, serine. And to that serine, through an O-link,
Go ahead.
is that mine?
You're at 5721 USA North Drive.
Sorry. Mind of its own. Alright, so we have peptides, right, a protein structure, and attached to the O of the serine is your sugar group, right, specifically serine. What else amino acid do you think of can have a group attached to it? What else? Can't forget it. Yeah, threonine. I think I put that on a test. Serine and threonine are similar. Okay, good. So you might wonder, why not tyrosine? It has an OH group. And about 10 years ago, they found actually a protein in bacteria that has a tyrosine attached to it and it's actually gotten glycosylated. And the only one known to date for humans is glycogenin, and that protein has a tyrosine that has been glycosylated. Alright? But I'm sure there's plenty, they just haven't discovered it yet. But it's mainly serine and threonine that they've seen. Proteins that have asparagine in particular have glycosylation groups attached to it. And it's through, here's your asparagine, there's nitrogen, right? That's your R chain, nitrogen is here, and it's linked to a sugar group, and this is your N linkage. Okay, so you've seen now O linkage, and you've seen N linkage as well. So those are different types of glycoproteins, and glycoproteins, when you look at glycoproteins specifically, there's more protein by mass than there is carbohydrates. So that's a main difference. Okay, so here I'm showing you patterns. We used to memorize these. Don't ask me what I memorized because I don't remember them. So I'm not going to torture you by having you memorize it. But there is an importance to it. And when I talk about blood groups and what they know about the blood groups, and from there the enzymes that can actually cut the different carbohydrates from those blood groups are serving as a source of research. And I'll talk about that in a minute. So asparagine has a pattern, right? So it has sugar groups here and branches out with different mannoses. So the whole take home is it's very specific. Erythropoietin is an example of a glycoprotein. And so by mass wise, there is a lot of protein and some glycosylated versions of either serine or asparagine. So they are secreted by kidneys into the blood that stimulates red blood cells production. And so it actually enhances the stability of that protein of glycosylation. So athletes may take man-made erythropoietin, pharmaceutically made erythropoietin. The way they discover that it is pharmaceutically taken and it's not the original is by the patterns. Okay, so the pattern of the normal that we have is very different from the one that is made pharmaceutically. Why is it different? Why can't we make it the same? Because it's not stable this way outside. So they make it so that the structure is similar, but it's not identical. Because the glycosylation patterns are different when you actually synthesize it. So they can detect two patterns, whether it is a normal erythropoietin or one that has been taken. Alright, so that's glycoprotein examples of glycoproteins. So next is proteoglycans. Proteoglycans are a little bit different because they're very specific by having a lot more carbohydrates than protein by mass. Right, so that's a difference. So the type of protein is not as much, but the carbohydrate by mass is greater. The carbohydrate is called glycosaminoglycans or GAGs. So they are repeating units, it's very specific. They have two types of amino sugars, and they have a negative charge associated with it. So when the protein has that type of a glycosaminoglycan, it automatically is grouped into a proteoglycan family. You'll see them in cartilages, and I'll show you that in a minute. So they do actually serve as an extracellular matrix and serve as lubricants. So these are my two examples of the glycosaminoglycan structures, the glucosamine and galactosamine. So all proteoglycans will have a combination of these two, either of these two. They can be just glucosamine or just galactosamine, or they can be a combination. And then on top of that, they will have charge, one or more carboxylate or sulfate groups, and negative charge, very specific negative charge. So you can see that here, you've got, let's see, I think I talked about it a little bit about, it looks like ferric and sulfur, so let's use that. So here are the two, glucosamine and galactosamine attached, and then attached to it is a sulfate group. So when you see a structure like that, it belongs to the glycosamine glycan family, and that is attached to a protein, and automatically that group becomes a proteoglycan. Okay, so with the cartilages, you'll see those types of protein. And why are they important? Because of the negative charge that's associated with the glycosaminoglycan. So because they have the negative charge, if you hit your foot, that water starts to be absorbed by the negative charges, and it protects your foot. So it acts as a lubricant. So there's a significance for why certain types of proteins are located in certain areas. So mucins are an interesting one. They are mostly O-glycosylated, and they are present in different forms actually. Cancer cells, the regular mucins are present, but then the patterns change in cancer cells. And when those cancer cells change the patterns of the glycosylation, they become more oncogenic, which means they become more cancerous. These mutated versions that have these mucins that are classified as oncogenes or have gone through these mutations in terms of the patterns get overexpressed in tumors. And the reason for these examples of having carbohydrates, you wonder, why do we have these carbohydrates as decorations? Well, number one, the mucins are able to communicate and migrate, the tumor cells are able to migrate from one organ system to another by this cell-to-cell interaction. So these carbohydrates groups will migrate towards another area that has a fine binding pocket and will attach itself. So that's how mucins work. They allow the progression of the disease to occur by metastasizing, going from one organ system to another. Okay, so like I said, glycosylation, the patterns are important. And we've seen that with erythropoietin. Another thing that I think is really interesting is pretty new, about 2019 to 2020. They discovered in bacteria an enzyme that can actually cut groups here, right here. They can cut here. And they can actually cut in bacteria specific bonds that have sugar groups attached to them. So they've isolated this, and what they're trying to do is try to see if we can actually get a lot more O blood group by using this enzyme is beginning to cut at specific groups. Notice that the O and A and B antigen are specific because of the glycosylation patterns. Right? So these antigens are post-translationally modified, and with those patterns, you can tell whether it's a B, A, or an O. To get a universal donor like O antigen, they want to use this enzyme that they have discovered from bacteria and begin to take the A or the B and begin cutting off that pattern of sugars so that it all becomes O. Give me right? So obviously, someone with an A antigen can take up the O antigen because the patterns are not there, right? But it can take the B because it's different. Okay, so that's where things are going, and I think I have, there's a magazine here, Science Magazine, published this about a couple of years ago. So you can read up on that. All right, so any questions so far? You're good? Okay. All right, so let's look at peptidoglycan. Why am I talking about it here? I'm talking about it because the cell wall has sugar groups attached to it. So it protects the outside from the inside, and this is your peptidoglycan within the cell wall. So the name says it, right? The peptidoglycan, it has peptides, which means it has amino acids, and it has a glycan, which means it has carbohydrates groups. So it's a combination of amino acids and carbohydrates that make up the cell wall structure. The names are not important, so don't memorize these, but these are the sugars. So they alternate between N-galactosamine and N-muramic acid. So these are the different types of sugars that are part of the cell membrane. And so you see them repeating. So there's your polysaccharide, right? So these are the repeating sugars, that's your polysaccharide. Cross-linked to the, here's my pentaglycine. So polysaccharide, and then it's cross-linked with a tetrapeptide and a pentaglycine attached to it. So the pentaglycine is obviously 5 glycine molecules. The tetrapeptide is very important. Tetrapeptide is a repeating unit of 4, and it repeats itself. So let me show you what that is. So here's your pentaglycine bridge, right? And connected to that is these 4 different amino acids. Alanine, isoglutamate, lysine, and alanine. And on the bottom, like I said, it repeats itself. Alright, so you'll have alanine, isoglutamate, lysine, and alanine, and it repeats. Okay, so these tetrapeptides repeat. Okay, so let's just look at this part here. Alanine and alanine. Alright, so it's linked together. 2 alanines are linked together. And then look at that structure, and they created penicillin. And if you look at the penicillin structure, it looks pretty similar, right, to the alanine-alanine structure. Okay. So here's your part here, one part here, and the other part is here. So here's the alpha carbon, nitrogen, carboxylic acid, and it forms a ring structure here. Here's the peptide bond, alpha carbon, nitrogen, carboxylic acid, and then it forms a ring. So penicillin was created based on what they knew about the amino acid structure that's found within the peptidoglycan. So the enzyme that creates the entire peptidoglycan is called transpeptidase. Okay, so that is the enzyme in the bacteria that creates the cell wall peptidoglycan. So the transpeptidase, instead of taking and incorporating the alanine and the other amino acid, it accidentally incorporates the penicillin, and when it does that, you can't build a cell wall, and it breaks down. And that's how penicillin works. Okay? Structural similarity. And so next semester, we'll talk more about the reactive sites, a little bit more about the reactive sites of beta-lactam. And and how bacteria become resistant to different types of antibiotics like penicillin that have a beta-lactam load. So we'll talk about that next semester. But transpeptidase, because it's voluntarily taking that penicillin, they call penicillin a suicide inhibitor. Okay, so that's your reaction, that's irreversible. The penicillin binds specifically to a serine molecule within the transpeptidase, and in doing so, you prevent that building of the peptidoglycan. Lectins are really nice. I've always wanted to do some research on lectins, but I never got the chance to do it because they have carbohydrate binding proteins. They can bind to them. So they contain 2 or more binding sites. Okay, so these are proteins that have pockets in them that bind to other proteins that have carbohydrates or carbohydrates alone. So here's your glycan binding protein. That's your protein. That's the pocket. And it binds to carbohydrates. And these carbohydrates can be associated with another protein, or they can be just associated with the cell membrane. Alright, so here's that interaction, that weak interaction that occurs. Why do we have these types of lectins? Well, they facilitate cell-to-cell interaction. Just like mucins, they're able to migrate and attach, but mucins have carbohydrates, right? They're not lectins, they have already carbohydrates and they migrate. Lectins are different because they don't have carbohydrates, they have pockets for where the carbohydrates will bind. So they're able to still migrate. Where do you see such things? In immunology, if you study any of those, selectins are a class of lectins, and some of the selectins bind. They're part of the immune system, like lymphocytes, for example. So lymphocytes have lectins attached to them, and then they're able to migrate towards the site of injury that have carbohydrate groups and attach to them and cause inflammation. Other places is attachment. Embryo attaches, placenta, uterus. So the embryo has lectins, and it can attach to the mother's uterus by interaction with the carbohydrates. Okay, and it implants itself. Okay, so the last little bit that I want to talk about is sialic acid and Tamiflu and influenza virus. So here you have your virus, and the virus has some clinical features to it. It attaches to the host. We all know that it attaches to the host, but it's very specific, non-covalent interactions that occur. So it has this protein called hemagglutinin, and it recognizes specific sugar groups called sialic acid. So when the host has, there's your protein here, this particular protein has sugar groups attached to it. And so this could be what? A protein like this attached to sugar group could be an example of a glycoprotein. Right? Glycoprotein could be an example of that. So this protein that has a glycoprotein that has specifically sialic acid can be recognized by the hemagglutinin on the virus, and it attaches. And then what will happen is the viruses will go into the cell and will start replicating, transcribing, translating, and so forth. So the action, really, of the virus is not to just stay in one spot, right? It wants to migrate and start to proliferate in other areas and grow and infect. So this enzyme, called neuraminidase, is like a little scissor that detaches hemagglutinin from being attaching to the host. So the neuraminidase is also expressed on the virus. When it's expressed, it'll go to cut between these two interactions. And now the virus is free to move to another distal organ system and infect. Tamiflu, what it does is it's very specific. It's going to block the activity of neuraminidase. So that's why it's recommended to take it within 48 hours before that virus cuts and begins to migrate to other places. Because after that, it's useless, right? After that, you just have to rest and take care of yourself and get through it naturally without the drug. Alright, do you guys have any questions about this? And so here you see the attachment here. Once it attaches, it goes in, the virus particles are replicating. You have neuraminidase, it cuts, and then starts to move. So the hemagglutinin would be an example, because it's binding to a carbohydrate. It's an example of a lectin, right? And that's the example of a lectin. A lectin is a protein that has binding pockets for carbohydrate groups. So here, with the virus, hemagglutinin is a lectin, and it binds to sialic acid. What can the virus do to prevent itself from, people become resistant? How can it do that? Well, the virus can mutate. The virus can mutate the neuraminidase structure, right? It can mutate that. Just change the primary structure and it changes the protein structure. The virus can also now have a different type of hemagglutinin that recognizes something else, not specifically sialic acid, but some other carbohydrate group that Tamiflu can no longer work on. Right? So those are different ways the virus can help itself to be able to replicate and not become resistant, not become killed by the Tamiflu. All right, so we do have some time. Do you guys have any questions? All right, I do want to get started on the hemoglobin. So let's work on that. Give you about 5 minutes or so to work on the first question.
Anybody need a sheet?
What'd you get?
Anyone else?
The only one I didn't get was 3. Figured it out. Ours can do them. So it just ended up. I don't know, I wonder why. Was it good? Because I don't know.
Do you have a quiz today?
No, not until
No.
next week. That just gave me
I think you have an assignment, right?
It's the same time as the quiz.
Is it?
That's next week, I think, isn't it? Yeah, next week Tuesday. And then we have a
Okay,
second.
oh, we're talking about the due date. Same
Yeah.
time the assignment is due, that is.
Yeah. I think I know the answer, but might as well say sorry. Thank you. But you don't get to. Yeah, I am. I almost brought you out, but. Me too. I parked in not spot. But it's when I feel like off cupcakes and I'm in charge of picking up cupcakes. So like flower girl.
Because the next one's actually off, right? So it's just, you know what? there, not where I was. So we don't. Okay, so it's here. Where you got 50, right? Okay, so it's there. That makes more sense. I was asking for like high temp pressure at 50. Okay, so apparently 250 is what it feels for like the center. Oh. Wouldn't that be kind of the last if you would take one? Yeah.
No, I meant for number 2.